Hi, welcome to the Bio and AD Tutoring Center videos. Today we're going to be talking about the citric acid cycle. It's also called the Krebs cycle. So before we actually get to the actual citric acid cycle, we have to talk about a basic step first. It's called pyruvate transport. And I didn't have enough room on my board, so I put it on this mini whiteboard, so hopefully you can see it. Glycolysis, as we know, generates 2-pyruvate. The pyruvate isn't the molecule that can just enter right into the citric acid cycle. It has to be converted first. And this process is called pyruvate transport, and it occurs as the pyruvate is entering the mitochondria from the cytosol, because remember that's where glycolysis occurs. So as it enters, it loses the CO2. So pyruvate is a 3-carbon molecule, if we remember, because we took glucose and essentially broke it into 2 molecules. So pyruvate is a 3-carbon molecule, and so when we lose this CO2, our acetyl-CoA at the end is going to be a 2-carbon molecule. And then what we also do is we take our coenzyme NAD+, and we change it, we reduce it to NADH2 by stripping this pyruvate of electrons. And then the last thing we do is we add a coenzyme A onto the molecule. So then this acetyl-CoA is the substrate that is going to enter the citric acid cycle. So an important reminder, if you forgot a little bit about NAD plus and NADH2, that's kind of important for today's video. So just remember that NAD plus is a coenzyme, and it oxidizes these carbon molecules, so it strips them of electrons, so then it is reduced, and then it becomes an electron carrier, and it travels to the electron transport chain, which we're going to talk about in another video, and it donates its electrons to it, and then the energy from donating and transferring those electrons is ultimately used to generate ATP, which is the goal of cellular respiration. So the citric acid cycle, it happens in the mitochondrial matrix. So it might have been a while since we talked about organelles, but it's that interior part of mitochondria. And then the goal. So we're going to be talking about a lot of different processes and products and reactants, and sometimes it's hard to keep straight. But if you ultimately keep the goal of this process in mind, it will help you. The goal is to ultimately oxidize these carbon molecules to generate NADH2 and FADH2 so that they can be taken to the electron transport chain and used to make ATP. So we start off with acetyl-CoA. Acetyl-CoA is going to lose its coenzyme, and it's going to become citrate, this intermediate. Citrate is then going to lose a water and then get a water, so no net gain or loss of water. And in the process of losing and gaining, it's going to become isocitrate. Isocitrate is then, and so this was a 4-carbon molecule, this is a 4-carbon molecule, and we said this was a 2-carbon molecule from pyruvate transport. So when they come together, they are a 6-carbon molecule. So we're going to be counting carbons because that's important. So isocitrate is also a 6-carbon molecule, and isocitrate is going to lose a CO2, and it's also going to change on the side so you can see better, an NAD plus to NADH2. And when it does that, it's going to become alpha-ketoglutarate. And so because alpha-ketoglutarate, it's going to have 1 less carbon because we lost a CO2, so it's going to have a 5-carbon molecule. Alpha-ketoglutarate is going to do a very similar thing. It's going to lose a CO2, and it's going to change an NAD plus to an NADH2. And it's also going to add on a coenzyme A. And when it does that, it becomes succinyl-CoA, which is a 4-carbon molecule because we lost another CO2. Succinyl-CoA is going to lose that coenzyme A. It's going to gain a phosphate, and it's going to use that phosphate to change GDP to GTP, which is just another molecule that can be used to generate energy like ATP, and a lot of times this GTP will actually be used to make ATP. And so the making of ATP using substrates is called substrate level phosphorylation, and making ATP in the electron transport chain is called oxidative phosphorylation. So this is the one example where we make ATP via substrate level phosphorylation. We also do it in glycolysis. So now succinyl-CoA with all of this is going to become succinate. And so succinate is going to convert an FAD to an FADH2. And FAD is just another coenzyme, a lot like NAD+, it's just an electron carrier. And then fumarate is going to add an oxygen. A water. You can kind of remember that, like fumarate, fumes, it's fire, it's hot, so it wants water. That's an easy way to remember it. And it's going to change into malate, and then malate is going to do another NAD+ to NADH2. And this whole time, because we haven't lost any CO2, we're sticking at a 4-carbon molecule, which is where we get oxalate, another 4-carbon molecule. And then oxalate will combine with another acetyl-CoA, and the cycle will continue. So this cycle can sometimes be a lot to memorize, and it gets confusing. So what I recommend is going into an empty classroom somewhere and getting a whiteboard and a marker and just writing out a couple times, or even on a piece of paper, because writing it out a couple times really helps you memorize it. And if you're asked questions on a test about specific reactions, that's important. But even more important than knowing these individual steps is just knowing the net products of this. So let's write them out so that we can get them straight. So how many, we're going to start by doing this just with one acetyl-CoA, and then we'll go from there. So how many NADH2s did we produce? NADH2s, we produced 1, 2, 3, 4. So we produced 4 of those. I think you can see that. And then we produced only one FADH. Oh, sorry. This is FADH2. So this is 1, this is 3. And then CO2s, we produced 1, 2. And then ATPs, it's hidden behind my board, but we produced 1 ATP. Okay, but let's do this, the products of just one pyruvate molecule. So let's include pyruvate transport in this. So if we include that, then this is going to change to a 3 because we have another CO2 there. And this is going to change to a 4 because we have another NADH2. Now remember that glycolysis takes one glucose molecule and turns it into one pyruvate. So a lot of times on a test, they'll ask you how many NADH2s are made from one glucose molecule, and one glucose molecule makes 2 pyruvates. So all of this has to be doubled because it's going to have pyruvate transport twice and the citric acid cycle twice. So this number for one pyruvate is actually going to be 8, 2, 6, and 2. And now if you notice, we have 6 CO2 that are produced, and there's nowhere else that we're actually going to produce any more CO2 besides the process we talked about. And if you remember, the net equation for the cellular respiration is we take glucose, 6 water molecules, and we change it into 6 CO2s and 6 waters and ATP. And so this is where the 6 CO2s come from in that net reaction. So that's kind of nice to see. And so ultimately, this is just going to go off as a byproduct, the CO2. The ATP is just ATP we generated that we can now use. But now we have all this NADH2 and all this FADH2 that we can send to electron transport chain, where it can now be used to generate a whole bunch of ATP via oxidative phosphorylation. So I'll go more into oxidative phosphorylation in another video. I hope this was helpful, and have a good day.